Hi everyone, and welcome to today's lesson on enzymes and enzyme activity. The first thing I want to talk about is the brief introduction to enzymes and the concept of catalysis. So first we usually ask what is catalysis, which is increasing the rate of chemical reactions. And when we talk about enzymes, they are a form of a particular type of catalyst, specifically biological catalysts. Okay, so catalysts are the umbrella term for any of the structures that will help increase the rate of chemical reactions. And then enzymes are specifically biological catalysts. Now, most of the ones that you'll encounter in lessons are globular proteins, meaning that they have more than one subunit. But the exception that I want you to remember is self-splicing RNA. And that'll come back in future lessons, especially in a lot of your other courses that you take. Now, when we talk about enzymes, it's important to understand their action, meaning how they work. And some of the points that I want you to remember is that, first of all, we talk about catalysts helping to increase the speed of reactions, right? And so the way that enzymes do this is by lowering what's called the activation energy of a reaction, and activation being the starting of that reaction, right? So activation energy is the minimum amount of energy required for a chemical reaction to occur. Now note that this does not change the overall free energy, which you see as delta G in figures like this one. The free energy is the difference in the energy between reactants and products, okay? So when you're looking at any reaction graphs, the peak that you see in the graph, so the peak up here, the peak up here, that's the activation energy, the amount of energy required to start that reaction. And you'll notice without an enzyme, that peak is all the way up here, but then with an enzyme, it's much lower down here. And if you lower that activation energy, it means it's now easier or quicker to start that reaction. And the example that I like to give for this is, let's say you're planning on buying a new phone, right? And when you want to buy that new phone, let's say it's $1,000 for this beautiful fancy smartphone that you want to get. Now, saving up to that $1,000 is basically your activation energy when you'll get to finally start to use that phone once you reach $1,000. Now, what happens if they put that phone on sale while you're saving up and now it's only $100, right? Yeah, I wish. But for this example, let's say suddenly it goes from $1,000 to $100. Well, now you're going to be able to get that phone much quicker and start using it much quicker because it's a lot easier to save up $100 than it is to save up $1,000. Okay, so that's basically what enzymes do in terms of why they make the reaction happen quicker. Now, there are a lot of things that can affect the rate of a reaction, whether there's an enzyme or not. And one of the big ones that you tend to hear about is the effect of temperature on a chemical reaction. Now, it makes sense that in general, the higher the temperature is, or the more you raise the temperature, the faster a reaction is going to occur, right? Because higher temperatures increase the speed of molecules and how they're vibrating and bouncing around. And if you increase that speed, then they're more likely to hit into each other and react with each other. Now, when we talk about enzymatically controlled reactions, yes, raising the temperature will make the reaction go faster, but only to a certain point, as you can see in this graph. So the reaction activity will increase as the temperature is going up. But you notice at about 50 degrees, now the activity of the reaction decreases instead of getting faster. The reason that is what happens to enzymes, which are proteins, at high temperatures? Well, they denature, right? So if the enzyme's denaturing, well, now you are going to stall or stop that reaction from occurring. So above a certain temperature, the reaction will no longer go faster. Instead, it will go slower and the activity may completely stop. Okay, so I want to take a moment to show you a clinical usage for enzymes. It's actually pretty cool that enzymes can be used as markers for disease. And so what we do, in order to understand this, it's important to know the term isozymes. And isozymes simply means the different forms of an enzyme, meaning you have the same enzyme with the same function. So here you have LDH, which is lactate dehydrogenase. You don't have to know the full term. Knowing LDH is fine. So here you have LDH, the same enzyme 5 different times. But as you can see, it has slightly different forms. So notice each one has a slightly different subunit change compared to the previous one. Okay, so these 5 versions of LDH are isozymes, the same enzyme with the same function, but with slightly different structural forms. Now, what's important about isozymes is we can use them to help us diagnose different medical issues. So for instance, in the example of this LDH, this lactate dehydrogenase, because it can exist in those different forms, you'll notice that the different forms can be in different locations of the body and can indicate different things. So for instance, if you find an increase of LDH or the LDH isozymes in heart muscle, then you can diagnose that the patient most likely had a heart attack. Whereas if you find it in the blood, that may show a different kind of tissue damage. Now, similarly, there's another enzyme called CK, which stands for creatine kinase. And with the creatine kinase, that's an enzyme that can occur in different places as well. It can be in the brain, the heart, and skeletal muscles. And depending on where you find CK and its isozymes, that will help you diagnose different things. So if you find the enzyme's isozyme in the brain, well, then that can signal that there's been a stroke or brain tumor. Whereas the heart type, the heart version of the isozyme, would instead indicate a heart attack. Okay, so kind of where you find the elevated levels of an isozyme and which version you find can help us diagnose different diseases. Now, stepping away from that clinical aspect, I want to get back into the enzymes themselves, including reactions. So I'm highlighting over here. This is to help show you how we can write out a reaction. And in that reaction that you see here, the 3 different letters, I've put what they mean over here. A and B are the reactants that then produce P, which is the product. Now, when we demonstrate the rate of a reaction, you're going to notice that you have different writings over here. You can demonstrate the rate of the reaction based on the disappearance of the products. So when you see a negative here and a negative here, that's the disappearance of that particular reactant. Sorry, not the product, the reactant. So the negative here is the disappearance of the A reactant. The negative here is the disappearance of the B reactant over a certain amount of time. So delta T means the change in time. Or you can represent the rate of the reaction as the production of the product. So you notice there's no negative here because instead of it being something getting used up, a positive indicates something being made, suddenly appearing, okay? So negative is the disappearance of the reactants. Positive is the appearance of the product. And again, This is showing the change in the product or the change in the reactants over a given amount of time. Now, when we talk about enzymes, their whole function and their ability to help a reaction occur is based on their ability to bind substrates. Now, when we talk about the enzyme binding a substrate, you'll notice that there are two models that will allow us to better explain how the enzyme binds that substrate and then ultimately releases the products of the reaction while not getting used up in that particular reaction. Now, the figure on the left here, this demonstrates the lock and key model of enzyme substrate binding. And so as you can see, the shape of the enzyme's active site, which is highlighted in green here, is a very specific identical match to the shape of the substrate, which is this little blue pizza pie shaped structure here. And so you see that their perfect match fits right in, and nothing is changing in terms of the shape of the enzyme. The alternative model is the induced fit model, which you see over here. And so this big glob of an enzyme, you notice it starts off having these gaps because it was not, the active site was not a perfect fit for the substrate. And instead, what happened was that the active site underwent a slight change in shape to better fit the substrate. Okay, and that's currently the most accepted model, but depending on the enzyme and depending on the substrate, both models still exist. So lock and key, the substrate and the active site are a perfect match with no change in shape to the enzyme, whereas induced fit, there is not a perfect shape match, so the enzyme undergoes a slight change in shape first to properly bind that substrate. Now, since we just mentioned the enzyme's active site and the binding of the substrates, I just wanted to give you a brief reminder of how enzymes actually work, and there's an example that I always like to give to students. So basically, like you always hear, enzymes help speed up a reaction, but are not actually part of the reaction. They are not used up in it. What they're basically doing is they are providing a seat or a location to put the substrates in close contact with each other, close proximity, so that those reactants can then react. Because for a reaction to occur, things have to be close together. And the example that I always use is the idea of a school bus seat. Let's say that students are going on a field trip, students that had never met before, and when the students get onto that bus and you have two students sitting next to each other on that seat for a long amount of time, let's say an hour-long ride, what's going to happen? Because they're sitting close together, they're going to start chit-chatting and talking. And by the end of that ride, they're now going to leave that bus basically going, oh my God, we're best friends, because they have bonded, right, by being close together. And that's what an enzyme does. It provides that seat. And just like with the school bus, well, what happens when those students leave? That school bus seat doesn't disappear. It hasn't been used up. No, it's there for the next set of students to then come in, sit down, bond, and leave. Okay, and so as you see in this figure, that is enzyme action. It provides like a seat for substrates to bind to the active site, be close together to each other, have a bond formed, or in some cases have, a reaction in which a product is made where something's been broken. And then the enzyme goes off to be used for the next reaction, having not been used up, having not been changed, as it is not actually part of the reaction itself. Okay, so now we're going to head into the not so much fun stuff, which is the idea of enzyme kinetics. The first thing we talk about is the Michaelis-Menten approach to enzyme kinetics, and there are some equations and some letter representations that I want you to know what they mean and be able to recognize and identify them. So the first thing that I'm going to show you is how we demonstrate the conversion of a single substrate to a product. So that's when you see S to P, this stands for a reaction in which you see a single substrate going to product. Now, what you see down here is how they demonstrate the mechanism for an enzyme catalyzed reaction. And I don't want you rushing to try and jot down what I'm about to say. What I will do is the next slide that I added to this video has a recap of each of the letters I'm going to tell you about, so you don't have to worry about writing them down just yet. You'll be able to pause the next slide and be able to see them written out for you. So when we're looking at an enzyme catalyzed reaction, which is the one down here, when you see the letter E, this represents an enzyme that does not yet have any substrates bound to it. Okay, so this is just the enzyme, and the S is just the substrate. And when you put them together, well, now you have an enzyme substrate complex. So whenever you see ES, it means that an enzyme has substrates currently bound to it. Now you'll notice this K1 and K-1, both as subscripts. K subscript 1 here, that is the rate constant for the formation of the enzyme substrate complex. Okay, so the rate constant for forming this structure right here. We said earlier that when you see a negative, that usually means the reverse of a reaction, something, you know, breaking apart, for instance. And so that's what you see here. K-1 is the release of the substrates from the enzyme. So that's the rate constant for the release of the substrates from the enzyme. Also, you'll hear it called the dissociation of the enzyme substrate complex, so that it's back to being a free enzyme and the free substrate. The second half that you see here is the enzyme substrate complex allowing for the production of the product. And so you have another rate constant, which is K to the subscript 2. That's the rate constant for the conversion of this enzyme substrate complex into free enzyme and free product. Okay, so it's the release of the product here. Okay, so picture an enzyme and a substrate binding together in the substrate binding the active site. You now have a single complex, and then once the reaction has occurred, these substrates are now a product that has been released. As I promised you, here is a recap slide. So here I listed out each of the terms that I mentioned on the previous slide, and I show you exactly what they stand for. So you can pause this slide and jot down all of your notes. Now, the textbook definition of what Michaelis-Menten kinetics are is that the Michaelis-Menten kinetic is meant to describe enzymatic reactions where a maximum rate of reaction is reached when substrate concentration achieves 100% enzyme saturation. Okay, now as we go through the next few slides, this concept of what exactly this approach is and what the terminology is will become more and more clear. So you don't have to memorize that definition. I just wanted to, let you know what basically textbooks will tell you as they introduce you to the concept. Now, the big equation that you tend to see with the Michaelis-Menten approach is right here. They give you what each of the terms within this equation mean. So you have initial velocity over here being equal to the maximum velocity over the substrate concentration at half V max, which we're going to see in graphical form in a minute to help you better visualize what that means, plus the substrate concentration. Now I want you to add a little note that this Km, sometimes you'll hear it called the Michaelis constant. Okay, so this is how you spell Michaelis, and it is called the Michaelis constant, which is basically a number representing the strength of binding of a substrate to an enzyme. And so that's going to be very important. So put a star next to the Km, letting yourself know that sometimes it's called the Michaelis constant, and it is a number representing the strength of binding of a substrate to an enzyme. So now you're going to get a little better visual of some of those terms that we saw in the previous slide. When we're talking about this equation, you'll notice the graph here for the enzyme reaction. We have two different aspects of this hyperbolic graph. We have first order kinetics, which on this graph is right here, and then we have zero order kinetics, okay? And over here I ask you, what's the difference between them? So I want you to know two things. First of all, I want you to know where you find them on the reaction graph. So first order is over here, and then zero order is where you have the flat lining of the hyperbolic curve, okay? The second thing I want you to know is what they actually mean. So this first portion that's increasing, it's increasing because first order kinetics means that the rate that you see here depends on the concentration of the substrate. Okay, so the reaction at this point is rate dependent on concentration of substrate. And then at the point of zero order kinetics, the rate no longer depends on the concentration of the substrate. Okay, so basically in the lower region of the curve, the graph that you'll see, the reaction is going to start out first order, which shows that the velocity, which is V, okay, depends on the substrate, which is S, the substrate concentration, which is S, and then eventually the active sites of all of the enzyme molecules have become saturated, right? They're all saturated, meaning they're all filled up. And so now you end up with zero order kinetics, okay? You basically have the flat lining of the reaction, okay? And another way to think of this is basically at low substrate concentrations, which we just went over on the graph, first order kinetics are observed. Whereas at higher substrate concentrations, okay, then you have zero order kinetics observed. So, keep that in mind as one of the ways that you could be asked about this. You can be asked, at low substrate concentration, what form or what order of kinetics would be observed. And so low substrate is over here. Okay, high substrate is over here because substrate concentration is your x-axis. Okay, the figure also shows where you find the Michaelis constant that we mentioned previously, the Km. So it shows that when the rate of the reaction, okay, rate is the velocity that you see on the y-axis, when the rate of the reaction is half its maximum value, okay, half Vmax, that is when the substrate concentration is going to be equal to the Michaelis constant. Okay, so make note of this point here, that what you see here, when the rate of the reaction, okay, which is the y-axis, is half of the maximum value, half of the Vmax, that is when you see the substrate concentration being represented as the Michaelis constant. Now I want to show you some of the mathematical relationships that exist amongst the different terms that we've been using so far with the Michaelis-Menten information that we've been going over. Now, a big part of this is you should be able to recognize what some of these formulas actually mean if you were to see them, because this isn't an in-person class. It's not like you're going to be doing short answers, writing equations out. Instead, your exams with these questions will basically be multiple choice, so you'll have to recognize some of these equations and what it actually tells you. So the first one that you see here in terms of the relationships is looking at the enzyme catalyzed reaction of formation, meaning forming the enzyme substrate complex. Okay, so you're seeing the binding of the enzyme to the substrate to form the complex, which is the ES. Okay, and that will then form the products ultimately. Now, notice what we went over earlier. So when you look at rates and you're looking at, the change in certain things over a certain amount of time, which is the delta T underneath the rate equation, you'll notice that positive, just like previously, is a rate of formation, whereas negative now means it's the reverse reaction, meaning the rate of the breakdown. So breaking down that enzyme substrate. Okay. Now, when you look at enzymes, enzymes are capable of processing the substrates very efficiently, and that's basically their job. And soon what happens when you have these kind of reactions going on, the formation reaction and the breakdown reaction of this enzyme substrate complex, you eventually reach what's known as the steady state. And equation-wise, that's represented here, which now that you are pros and experts at these equations up top here, you know that in verbal lingo, what these equations are telling you is the rate of formation, which is this positive equation here, this positive fraction here, is equal to the rate of the enzyme substrate breakdown, which is the negative. You see the negative, so that's the breakdown. So in word formation, the steady state, what that means is the rate of formation of the enzyme substrate complex equals the rate of its breakdown. It can also be shown by the other side of the equations that we have up here, which is shown over here. This side represents the rate of formation is equal to this side representing the rate of breakdown, okay? But if I were to show it to you, I'd focus on this side here because it's usually a little more clear of what each of these is representing. Okay, so make sure you're able to recognize if you see one of these equations, the positive one is the rate of formation, the negative one is the rate of breakdown, and please know the definition of steady state. Now, there are three mechanisms to describe the order in which substrates can bind to an enzyme, because there are multiple substrates sometimes binding to this single enzyme. So you have these three different mechanisms. Notice that I color-coded the figures for you. So the blue term matches the blue outlined figure, red to red and purple to purple. And I'm going to have a recap slide come up next with the definitions that I'm about to give you. Okay, so you don't have to worry about trying to jot them down while you're looking at the screen right now. The first mechanism is called the ordered mechanism, and that's represented by this graphic here. Basically, it's an enzyme mechanism where the substrates have to bind to that particular enzyme in a specific order. Okay, so as you can see in this figure, the substrate A has to bind to the enzyme first, and then the substrate B has to bind to the enzyme second in order for that to occur. Okay, and then here you see P and Q as the ordered mechanism. Okay, so in the ordered mechanism, substrates have to bind in a specific order. Then you have the random mechanism, which looks a bit more chaotic because in this one, the substrates can bind to the enzyme in any order. Okay, so it would not have to be A, then B. It could be A first, it could be B first, it could be P, Q, whichever one is binding. It doesn't matter which one does it first. You'll ultimately get the same reaction occurring no matter which substrate is binding first. The last one is the ping pong mechanism, which you see down here. In the ping pong mechanism, that's where a substrate binds to the enzyme. It releases a product before the second substrate binds. Okay, so here you see the substrate is A, binding to the enzyme first. It then releases a product, which is P, before B, the second substrate, ends up binding, okay, to then create the next complex and next products. Okay, so ordered mechanism has to be a specific order. Random substrates can bind in any order. Ping pong is basically a sequential binding where a product is released first before the second substrate binds. Okay, and like I said, here is your recap slide. So this is a recap of the terms I just gave you so that you can pause and jot them down. Now, another way that we can work with enzyme kinetics is how you can calculate the Km and the Vmax from a graph. I know this slide is going to look a bit scary, but we're going to go through it in the next slide to show how it's actually done, and it makes it a little bit clearer. So if you are ever asked how do we calculate Km and Vmax from a graph, the answer is the Lineweaver-Burke method. And that's basically a double reciprocal plot, which you see here, graphical method for analyzing the kinetics of enzyme catalyzed reactions. And you notice I said a double reciprocal plot. Okay, so when you see 1 over V, that's the reciprocal of velocity. When you see, for instance, 1 over S, that's the reciprocal of substrate concentration. So that's why it's called a double reciprocal plot. And we're going to go over how you would actually do this in the next slide. Okay, so I said this does sound a little scary, but this is a textbook example of how to work with the Lineweaver-Burke method. So someone can be given this information here, which is the substrate concentrations and the velocity amounts for a particular enzyme-led reaction, okay, an enzyme-guided reaction, I should say. And what you notice when we talked about the Lineweaver-Burke method, we said it was a reciprocal, a double reciprocal. So the first thing that you would do is take the reciprocal of each of your columns. So remember, the reciprocal of substrate concentration is simply 1 over substrate concentration. So you would do 1 divided by 2.5. That would be your first point for your first x-axis plotting. You would have 1 over 5, which gives 0.2, 1 over 10, and so forth. Okay, these would be your x values. Substrate is x. Then you would do your reciprocal velocity, which again is 1 over each of your velocity points to give you all of your y-axis numbers. Okay, and then these dots represent plotting each of those points. So the first point would be 0.4 and 41.667. So you see here 0.4, 41.667. Okay, so that's that first point here. And you would plot each of these as the x value and the y value, just like we did 0.4 on the x and 41.667 on the y, giving us that plot. From there, you see that there is the trend line that has been created. And the way that you would work this is on the previous slide, you saw in the graph on the previous slide that the y-intercept, when you do a Lineweaver-Burke method, the y-intercept, which is right over here, that represents 1 over Vmax. Okay, 1 over Vmax, which is the reciprocal, because again, anything with the reciprocal is 1 over. And then the x-axis, you take the reciprocal of that negative x-intercept to get 1 over km. Okay, so looking at this graph, once you've plotted these values and made that trend line, what you do to get Vmax is 1 over the y-intercept, which in this case would be 1 over 12, because here the y-intercept is 12. That would give you a Vmax of 0.083 millimeters per second to the negative 1 power, okay? And then you would look at the x-axis and do 1 divided by the x-intercept to get the km of 6.45, because you have done 1 over this x-intercept, which is minus 0.155. Okay, and my handwriting may be a little sloppy because of the angle I have to write on the screen, but just so you have it in writing, the y-intercept is 12. Sorry, it's not picking up. So I will we'll erase that for now. I'll scribble it out. And what I'll do is on the next slide, I'll add a recap slide to show you the numbers that I just said out loud, because today the touchscreen does not want to work as well as it usually does. And as promised, here's the recap since the touchscreen wasn't letting me write it out for you. I want you to be aware of what each the x and y value represents when you're working with this kind of a problem. How you would get the Vmax, which is the reciprocal of the y-intercept, meaning 1 over the y-intercept. And then how you would get the reciprocal of the x-intercept to give you the km. So how do you get the Vmax? How do you get the km? It's important for you to know each of those. Okay, and you can look back at the previous slide to see exactly those numbers as the intercepts that we saw. Okay, and then that gives you, in this case, the amounts. And sorry, sometimes I accidentally say millimeters. I don't know if I did it the previous slide just because I'm so used to talking about that kind of measurement instead of millimolar. So I apologize if I did it. I can't remember whether or not I did. This is another one of those slides where I'm going to throw a lot of information at you. It's all very important, so don't worry about trying to jot it down right now. In 2 slides, I'm going to give you a recap slide with the notes for you to write down for this slide and the next one. Okay, so what I want to focus on when it comes to enzyme inhibition is that first we'll talk about reversible inhibition, meaning you can change it back from being inhibited to being active again. So the reversible types of inhibition are competitive inhibition, uncompetitive inhibition, mixed non-competitive, and pure non-competitive inhibition. Okay, so when you look at this figure and some figures that you'll see in textbooks, it shows it as 3, but in reality, non-competitive inhibition has 2 different forms. It has the pure and the mixed form. Now, the first form of inhibition that you see here, competitive inhibition, you'll notice that the inhibitor is binding to the active site of the enzyme. And since the active site is where the substrate is supposed to bind, this substrate can no longer bind there. It's kind of like if you want to park in a parking space, but there's already a car there, even when it's that little car that you can't see until you're just about to turn into that spot. That's competitive inhibition. Non-competitive, like I said, there are 2 different forms. Okay, in both of these forms, the inhibitor, you'll notice, is binding somewhere else on the enzyme. It is not binding at the active site, it's binding somewhere else and causing a change of shape of that enzyme, which we call an allosteric change. So this is the word allosteric. Allosteric change means a change in shape of an enzyme. So in pure non-competitive inhibition, the binding of that inhibitor has no effect on the binding of the substrate. Okay, it's just that catalysis cannot occur, but it's not actually having an interaction when it comes to the substrate itself. And this is basically very rare. What's more common is mixed non-competitive inhibition, in which the binding of the inhibitor does affect the binding of the substrate, which makes more sense. If you're going to have an inhibitor binding and changing the shape of the enzyme, then you're going to have an effect on the binding of the substrate. The last form of inhibition is uncompetitive inhibition, which sounds like a joke, but it's real. And that's basically a type of inhibition where the inhibitor can only bind to an enzyme substrate complex, not to a free enzyme. Okay, so for instance, in the other two, you'll notice that the substrate is not bound to the enzyme. Okay, the substrate is this blob up here, the teal colored blob. In this last one, the only time the inhibitor can bind is if the substrate has already bound to that enzyme. So this is the ES, the enzyme substrate complex. Now on here, I put a table, that represents each form of inhibition and how it affects the Km and the Vmax. And any of you taking the MCAT, this is usually a chart that they have you study as you prepare for the MCAT. So for instance, knowing that competitive enzyme inhibition will increase the Km but have no change on the Vmax. That's important, okay, knowing these two columns here. And you'll notice one of the ways to study this when you're prepping for the MCAT is you'll notice the Vmax decreases in every form of in every form of inhibition, except for competitive. That's where there's no change. And you can see no change. You could look at that C of change and remember competitive. The C of competitive is the one where there's no change in Vmax, all the rest decrease. Km is a little more difficult to study when you're prepping for the MCAT because, as you can see, some cases it increases, some cases it decreases, and in one case it has no change. Now, what I show you on this slide is we're looking at all four forms of inhibition, and we're looking to see how you can look at a graph, a Lineweaver-Burke plot, and tell right away which form of inhibition is occurring. Okay, and so what you do when you look at these graphs, you see the first one over here. That one represents competitive inhibitors. Okay, this one's competitive inhibitors, and the way that we tell that is that you see the curves intersecting on the y-axis. Okay, so in each of these graphs, you have the y-axis is this black line straight up and down here. You have the x-axis at the bottom of each of these graphs. In competitive inhibition, on the Lineweaver-Burke, the lines intersect on the y-axis. Then, when you're looking for a pure non-competitive inhibitor, you would then see the lines intersect on the x-axis instead. So you see the lines here now intersect on the x-axis when there's the inhibitor involved. Okay, that one is pure non-competitive, this graph right here, because it's the x-axis. The next one you can identify is a mixed non-competitive inhibitor. In that one, we're going to circle this guy here. This is the mixed non-competitive inhibitor Lineweaver-Burke plot. You can tell because the two lines intersect So when there's an inhibitor present versus when there's not, the lines intersect somewhere in the left quadrant. So this is the left quadrant, but you notice it is not directly on the x-axis. See how the lines are separated on the x? They intersect up here. That one is the mixed non-competitive. And then the very last one is usually the easiest to spot right away because you don't have to look for any intersection. This is the uncompetitive inhibitor. So if two lines are parallel on a Lineweaver-Burke plot, that tells you it is uncompetitive inhibitors, an uncompetitive inhibition in that reaction. Okay, so parallel on the Lineweaver-Burke plot. So please make sure I can tell you any of those intersections. I can tell you the lines intercept on the y-axis, on the x-axis, in the left quadrant, or the lines are parallel to each other. And depending on which one I state in a question about a Lineweaver-Burke plot, you should be able to tell me, is it competitive, pure non-competitive, mixed non-competitive, or uncompetitive inhibition. And you will notice that I put a recap here so you can pause because that was a whole lot of information. So these are the notes that I would like you to have on those two slides. In addition to the reversible forms of inhibition, there's also irreversible inhibition. And what happens here, as you can see in each of these figures here, you now have the inhibitor covalently binding to the enzyme. So it causes a permanent inactivation. Okay, so in this case, for instance, a heavy metal covalently binding to the enzyme so that it's permanently inactive. And in this case of penicillin covalently binding and permanently blocking one of the enzymes in bacteria for cell wall synthesis. Now, any of these substances that can covalently bind and irreversibly block an enzyme, we call them suicide substrates. So when you see the term suicide substrates, please know that that's irreversible. They irreversibly inhibit enzymes, and please know the examples that I give you. So in bacteria, penicillin is a suicide substrate, irreversibly inhibiting one of its enzymes for building its cell walls. And in humans, some of the examples of some really bad suicide substrates are cyanide gas, heavy metals, and nerve gas that will irreversibly block enzymes, and that's why they can cause severe sickness and death, because you need your enzymes active, not irreversibly blocked. Now, not all enzymes are Michaelis-Menten enzymes. There are also what we call allosteric enzymes. All that means is, as you can see in the figure here, so the figure over here, allosteric enzymes have an additional site beyond just the active site. And that additional site, for instance, can be a place for an inducer or repressor to bind. So here you see an inducer on this side, the inducer being an activator, binding somewhere other than the active site, and you see the inhibitor binding somewhere other than the active site. And you notice that that other site is called an allosteric site. The allosteric site means that binding something there will modify the shape of the enzyme. Now, there are two main ways for these allosteric enzymes to be controlled. One is the feedback inhibition, which comes up a lot in your biology courses. You'll see this in every class, cell bio, genetics, molecular, in our class here. Feedback inhibition, usually what you see is negative feedback inhibition. There's also positive ones, but we're going to focus on negative feedback inhibition. You see a pathway here guided by a whole lot of enzymes to make the ultimate final product down here. And in negative feedback inhibition, that final product is then an inhibitor that blocks an earlier enzyme, which then shuts down the whole pathway, which makes sense because at this point, you have the final product. So why would you want to waste resources making more and more of something you already have right now? So the presence of that final product blocks an enzyme earlier in the pathway. The other form of inhibition you see over here is similar to what we talked about in previous slides, where you can have inhibitors bind to the enzyme, make it so that the enzyme can no longer efficiently or effectively bind their substrate, and so now they have been inactivated. Now, as I mentioned in the previous slide, when you're talking about allosteric enzymes, you can no longer think of them the same way that we did with all of those previous non-allosteric enzymes. So like I said, Michaelis-Menten will no longer be used, will no longer apply to these guys. These allosteric enzymes, you're not going to hear competitive or non-competitive inhibition terminology. You're not going to see a KM. Instead, what you'll see are terms like K systems with a K 0.5 and V systems. Another difference you'll notice is now when we look at the plots for these allosteric enzymes, you'll notice it's no longer the hyperbolic plot. So before we saw a hyperbolic plot where you just had increasing and then flatlining. Now instead you have the sigmoidal curve, which is more like an S shape than the fully hyperbolic plot. Now, when you see K systems and V systems, all that means is a K system is an enzyme for which the substrate concentration that yields 1/2 Vmax is altered by the presence of inhibitors or activators. So I'll give a recap slide next for you to have the definition. And then in that system, the substrate level at 1/2 Vmax, now instead of being called Km, you see it as K 0.5, K 0.5. Okay, so that K 0.5 is the substrate level at 1/2 of Vmax in these allosteric enzyme curves. V systems, you also have V systems are combinations in which the allosteric enzymes and the inhibitors or the activators, when you have the presence of the inhibitor or activator, that will then change the maximal velocity of the enzyme, but not the substrate level that yields 1/2 of Vmax. Okay, so I know sometimes it's a little difficult when you're listening to someone say it, but I'll write out what I said and I'll write out the way the textbooks like to give it to you so that you can see it and put it in your notes to better understand it. Now, the key point when it comes to allosteric enzymes and the inhibition of these enzymes is the idea that binding to these enzymes changes the quaternary structure of the enzymes. So that means the binding of substrates, of inhibitors, of activators will change the fourth level structure, meaning the folded globular structure of these allosteric proteins. And as you've heard over and over in all your bio classes, structure determines function when it comes to proteins. So that's the key point with allosteric enzymes, is the binding of substrates, inhibitors, activators will change the structure of the enzyme, which then changes the behavior of that enzyme. That's the key point that I want to make with the allosteric enzymes. Okay, here's basically a transcript of the points that I wanted you to have in your notes for the previous slide, because I said a whole lot in that one little section. The next thing I want to talk about for allosteric effects, for allosteric enzymes, is the idea of allosteric effects. Now, one of the terms you'll see is allosteric effectors. That simply means any substance that will bind to the allosteric enzyme to affect its activity. So that can include substrates, inhibitors, and activators. Okay, so all three of those, substrates, inhibitors, and activators, write those down. Those are three examples of allosteric effectors. They will all be able to bind to these enzymes and affect its activity. Now, there are two categories I want you to know the difference between in terms of allosteric effects and allosteric effectors. There are homotropic effects and there are heterotropic effects. Now, homo in biology means same, hetero means different. So in homotropic allosteric effects, what you'll see is down here, all of the binding molecules. So we really see it in the bottom, in the rightmost picture here. All of the binding effectors are identical molecules. They're all the same molecule binding to that enzyme, usually at the same time, to produce an effect. At different sites, they're all the same molecule. Whereas heterotropic allostery or heterotropic effects is now when you have different substances binding to that same enzyme to create the effect on that enzyme. Okay, and so this can mean different substances being, one's an inhibitor, one's a substrate, for instance, both bound to that enzyme at the same time, okay, and having an effect. Because remember, we said the three effectors that I want you to know, substrates, inhibitors, and activators. If it's the same exact one bound to that enzyme, that's homotropic, and if it's more than one of them, different effectors bound at the same time, that's heterotropic. The next thing to look at when we're studying allosteric enzymes is to look at the principal models describing their behavior. So there are two models I want you to know. The top one here is the concerted model, and the bottom one here is the sequential model. Now what you notice in the concerted model, you have the enzyme that has different conformations. Whenever you see R, that's the relaxed conformation, which is the active conformation. Okay, so R is active. And then when you see T, that is the tight or also called taut conformation, which is the inactive conformation. Don't worry about jotting this down. I'm going to give you a recap of all of this information, so you can just look and listen for now. So whenever you see R, that's active. Whenever you see T, that is the inactive conformation, okay? And what happens in the different models, the concerted model, you have both of these subunits, so half and half, because, you know, enzymes are usually globular proteins, so they have more than one subunit. In the concerted model, the main aspect of this model is that the conformations of all subunits change simultaneously. So in this figure here, you'll notice that if, let's say, you have an enzyme that's in the R conformation, both subunits of that enzyme are in the R conformation, and both subunits change to the T conformation at the same time, and vice versa. If it's T conformation, both subunits are T, and they both change to the active R conformation at the very same time. Then in the sequential model, like the name suggests, instead of both of the subunits changing at the same time, you have a sequence of changes, meaning that this cooperative binding model shows that the binding in one subunit changes that subunit, which then induces a change in the second subunit right after that. Okay, so you notice it happened one at a time. First, the left-hand subunit changed to the active position, which then induced the right subunit, the right-hand subunit, to change to the active position. Okay, and that may sound kind of familiar to you because this type of behavior models the induced fit theory that we mentioned earlier. Now, as I mentioned before, that's a lot of information and some terms I wanted to make sure that you knew how to spell. So here is a recap slide for you to pause the video, write all of these notes down. Please make sure you know the difference between these two models. Please make sure that you know that the R conformation of an allosteric enzyme is the active conformation, whereas the T conformation is the inactive conformation. Okay, and please make sure that you know the sequential model is basically the induced fit theory as well. Now, when it comes to the sequential model, I want to point out that the sequential binding is not just with substrates, it also applies to the binding of activators and inhibitors. So both activators and inhibitors in the sequential model will basically follow the induced fit mechanism. And so as you can see with this figure here, this demonstrates that cooperative binding of inhibitors that you see in the sequential model or the induced fit model is, same thing. And that cooperative binding of inhibitors basically means that when the inhibitor binds to one subunit, so one half of the allosteric enzyme, it then causes the sequential effect that the other half of that enzyme will now have a lower affinity for binding the substrate, meaning it has been inhibited. And you'll notice in this model, when an inhibitor binds to the enzyme, because it's an inhibitor, it promotes the T state, because we said T state is the inactive form, okay? And it basically ensures that the other side will also stay as the T state, as opposed to if this picture was of an activator, you would see the R state promoted. Okay, so again, make sure you're comfortable with that terminology. The other thing that I want to mention is negative cooperative, sorry, this word always messes me up, negative cooperativity. My mouth does not like to say that word at all. Basically, that version of the sequential model is saying that the binding of the first ligand to any of these allosteric enzymes will reduce the affinity for the next ligand if this is a negative inhibitory type of ligand that's binding. Okay, so just make sure you're comfortable with these terms that have been highlighted here. When it comes to enzyme activity, one of the methods of control that you'll actually see come up in a lot of classes and in a lot of reactions is the idea of phosphorylation. Now, when we talk about phosphorylation, one of the terms you'll see constantly come up is kinases. So as you can see from the figure down here, protein kinases will catalyze the addition of a phosphate group to an enzyme or to a substrate, and phosphatases will remove phosphate groups from molecules, from substrates. Okay, so a kinase will add a phosphate group or catalyze the reaction that adds a phosphate group to a substrate, whereas a phosphatase will remove it. So in addition to having the definition of kinases up here, please also write phosphatases are the opposite, okay? They're responsible for the removal of a phosphate group from a substrate. And you can see in this figure here an example of the addition of a phosphate group. So here you have the phosphate group that was added to the serine residue over here at the hydroxyl area of that group. Okay. Now, when we talk about phosphorylation, unfortunately, it's not straightforward. It's not like, oh, phosphorylation will always increase activity, or, oh, phosphorylation will always decrease. Instead, phosphorylation can do both depending on the substrate and depending on the reaction. So in some reactions, you'll see that phosphorylation increases the activity of an enzyme, and in others it will decrease the activity. In fact, in some systems, the effect of two opposing enzymes are actually coordinated by phosphorylation. So one of the examples that your textbook gives is an enzyme in a catabolic pathway might be activated by phosphorylation, while at the same time its counterpart in an anabolic opposing pathway might then be inhibited by phosphorylation. Okay, but really emphasize these terms here and the fact that the activity can either increase or decrease. Another important term when you're studying enzyme is zymogen. A zymogen is an inactive protein that can be activated by specific hydrolysis of peptide bonds. So as you can see in the figures here and here, the two examples that I want you to know are trypsin and chymotrypsin. Okay, so that's how you spell trypsin over here, and chymotrypsin down here. Okay, those two terms, those are proteolytic enzymes that are the classic example of zymogens and their activation. So as you see in this figure, chymotrypsin is initially found or initially synthesized as chymotrypsinogen, which is the inactive zymogen. And then through hydrolysis, which is cleavage or cutting of bonds, breaking of bonds, you then end up with the active enzyme form. And in this figure here, you can see various digestive enzymes that are initially in an inactive zymogen form, and then through cleavage or hydrolysis, they are activated. So in each of these examples, that red color one is the zymogen that then gets activated into the proper enzyme. Okay, and so in the examples of trypsin and chymotrypsin, these are proteolytic enzymes. They're in the small intestine, is where you want their digestive properties, but when they're synthesized, they're actually synthesized in the pancreas. And so if these proteolytic digestive enzymes were active where they're formed in the pancreas, that would do major damage. So instead, you need them inactive as zymogens in the pancreas, and then in the small intestine, that is where they will be activated by the cleavage of their peptide bonds, like in this figure, and now they can do their job without doing damage. Another important example of zymogens, proteins that are first formed as inactive, that I want you to know are the caspases, okay, caspases. What those are is another important class of proteases, and they're responsible for many things in the cell, but one of the most significant is apoptosis, okay, and apoptosis, you've probably heard before, is programmed cell death, and that now gives you the answer to the second question that I proposed here, which is why is it important that caspases are synthesized as zymogens first, which you see in this structure here. So they're synthesized as inactivated, and in this case, dimerization puts them into their active form. So it's not like the previous slide where you saw cleavage putting them in the active form. For caspases, it's dimerization, linking the subunits together, that makes them active. And it's important that they're synthesized as inactive first because, well, we just said they could cause apoptosis. So you need that to be very, very, very controlled. Okay, you only want cell death in certain scenarios. So if they were floating around the cell in their active form from the beginning, that cell would then die. It would be unwanted cell death. Okay? And that would have severe consequences to you. Now, in certain diseases, illnesses, or problems, you can have scenarios with uncontrolled cell death. and the disruption of apoptosis, and it can be very problematic. Okay, so why is it important that they're synthesized as zymogens first? It's so that you do not have unwanted premature cell death. Now, one of the most significant aspects when we talk about enzymes is the active site that we keep mentioning throughout today's lesson. And so I just want to give you a little bit of information on the nature of the active site. The first question that I posed for you is what influences the mechanism of the active site in catalysis? And again, this is another one of those slides where you don't have to worry about jotting down my answers. I will give you a recap with the answers on the next slide. So the point that I want to make in terms of what's influencing the active site is the idea that whichever amino acid residues are present at this active site is critical to how that active site will be involved in catalysis. Okay, so we actually call those the critical amino acid residues, are the ones that are at the active site. So for instance here, serine and histidine are two examples of critical amino acids in the figures here for chymotrypsin. And the fact that they're critical amino acids means that they are at the active site of this enzyme, and they are heavily involved in the abilities at that active site. Now, which part of an amino acid is most significant for its abilities and its binding and where the protein will fold? The answer to that is the functional group. And functional groups are most significant because they're what really gives the reactive properties to amino acids. So for instance, having a hydroxyl group or a sulfhydryl group, or especially having charge groups. So if you have a positive or negative charge, that will affect what that amino acid then interacts with and how it can then act at that active site. The last two terms that I want you to know on this slide are nucleophile and an electrophile. A nucleophile is basically going to bond to sites of positive charge, okay? So either areas that have positive charges or that are electron poor, meaning not very many electrons in that area. And that's opposed to the electrophile, which, as it sounds, you know, electrophile, loving electrons, so it's electron seeking, they're going to tend to bond to sites of negative charges. They'll seek out electron-rich sites. Now, this slide here is the recap that I promised, which is basically a transcript of the notes that I said in the previous slide that I want you to write down. So you can pause and jot down your notes. Now this is another one of those slides where I'm going to say a lot of information, but don't stress yourself out trying to write it down, since I will write the notes for you as a recap on the very next slide. So you could just pay attention and not worry about jotting things down yet. So on this slide, I start off by asking, what are the most common chemical reactions involved? Okay, when we're looking at the nucleophiles and the electrophiles. And so the first reaction that you see here are nucleophilic substitution reactions, and that's shown in this demonstration right here. And I color coordinated this slide like previous ones, where the red reaction is here, the acid-base catalysis reactions are in purple. So this red reaction here, what nucleophilic substitution reactions are reactions in which one functional group is replaced by another as the result of a nucleophilic attack. And as we saw in the previous slide, a nucleophile is going to want to attack electron-poor atoms. So in this demonstration here, the ... Z, that's the nucleophile, and the X is the leaving group, because that nucleophile has attacked over here and caused the leaving group X here to be separated. Okay. The other type of chemical reaction is the acid-base catalysis reactions, which could be general or metal ion reactions. And you see reactions here that are examples of acid-base catalysis, the top one being the example of when it's an acid, a general acid catalysis, where you have an amino acid donating a hydrogen ion. And the second one here would be an example of the general base catalysis, because now you notice that you see a hydroxyl group present here instead of the hydrogen ion. Okay. Now, what I want to point out when it comes to the general acid-base reactions is histidine is an amino acid that can actually take part in both reactions because of the reactive hydrogen on the imidazole side chain that it has that dissociates at certain pHs. And that was seen in the chymotrypsin mechanism, where histidine is part of both acid and base catalysis. Now, you don't have to memorize, the chymotrypsin mechanism and all that. I just want you to be aware that some amino acids like histidine will be part of both the acid and the base forms of catalysis. And the other thing I want to note is there's also what's called metal ion catalysis, and that's basically because in the Lewis formulation, an acid and a base is defined as, the electron pair acceptor and the electron pair donor. And metal ions such as, magnesium with that little +2 charge, they make for great Lewis acids, electron pair acceptors. Okay, so that's just to distinguish the metal ion catalysis from the general acid-base catalysis. And this slide here is as promised. You can pause the video. It has all of the notes that I wanted you to take down that I just said in the previous slide, so you can pause and jot all of that down. One of the last two terms that I want to go over before you are done with my annoying voice for this lecture at least is the term enzymes. And enzymes are really cool because you can think of an enzyme as basically a combination of antibody and enzyme, hence its name. And the enzymes are antibodies that have catalytic activity. And the example that I want you to be aware of is the clinical use of enzymes in the treatment of cocaine addiction, which is fascinating because as you can see from this figure here, this is how cocaine usually works. You have dopamine uptake to basically prevent having dopamine constantly bound to receptors, constantly triggering a signal, and cocaine blocks that uptake. So instead, you get an accumulation of dopamine between a presynaptic neuron and a postsynaptic neuron. And when you have that accumulation, because dopamine's not getting re-uptaken back into the presynaptic neuron, when you have that accumulation, well, now look, you have dopamine getting stuck and accumulated on the receptors of the postsynaptic neuron, which means you are getting the constant high, basically. It's accumulating that dopamine response. And so what we do in terms of enzymes is you have an antibody with catalytic activity. So in this figure, you see what a regular antibody looks like. And this is a catalytic antibody where the figure shows when the antigen binds, you get the catalysis of the reaction that gives you antigen products. So down here you see, in the case of using enzymes for cocaine addiction, the enzymes allow you to take cocaine and cleave it into the antigen products, basically. Sorry, into its two subunit products. And these basically are harmless degradation products that will not block the dopamine reuptake. So this blocking of the dopamine reuptake can only be accomplished by the full cocaine molecule, okay, as opposed to when the enzyme then cleaves that structure and gives harmless degradation products. And once you have that, you no longer have the prolonged neuronal stimulus, and ultimately the additive effects of that cocaine will vanish over time, and it will allow you to ease the suffering of a patient dealing with cocaine addiction. The last terminology that I want you to know for today is the concept of coenzymes and cofactors. And so even though the textbook will just say the word coenzymes as the unit that you find this in, it's important to know both terms because you hear both of them in a lot of the courses that you're going to take. And so basically both of these terms are non-proteins that will help enzymes function. They basically help activate enzymes. And the enzyme is usually a protein. The coenzyme or the cofactor, like I just said, are non-proteins, meaning they are not proteins. The difference between them is the idea that coenzymes are organic, whereas cofactors are inorganic. And usually when I teach these concepts, I tell students, when you think of cofactors and you see that word factor, picture a factory. Factories full of metal, minerals, inorganic material, whereas the coenzymes are organic and they're usually vitamins. And this slide helps you to understand why you're constantly told the significance of vitamins and minerals in your diet, and why taking supplements such as magnesium or zinc is sometimes highly recommended, because these vitamins, these minerals, and certain metals, they are responsible for allowing your enzymes to be active and to function. And as you know, your metabolism is completely guided and controlled by enzymes. You wouldn't be able to have all of those chemical reactions in your body, in your cells occurring without enzymes, and you wouldn't have enzymes properly functioning without coenzymes and cofactors. So please make sure you're comfortable with both of these terms and the examples that I put in parentheses for each of them. And that is it for today. If you have any questions, please contact me in the Remind app. I wrote the code on this slide for you to see that all you do is download the Remind app. You type in that code as your class code, and it allows you direct access to me. Basically, it will give me a notification on my phone, and it allows you to message with me in a way that's more easily accessible than emailing for a lot of people. And like I always tell my students, I have no life, so feel free to contact me anytime, day or night. If it's even the middle of the night when, some students wait till 1 a.m., 3 a.m. to watch the lessons, don't ever hesitate to send me a message. It won't wake me up, it won't bother me, but it'll prevent you from forgetting later on to ask that question. Okay? Thank you and have a great day.